Photosynthesis is an anabolic, endothermic redox process. It is a system that couples incoming light energy to the reduction of carbon dioxide into complex carbohydrates. This is the standard equation taught in biology. Carbon dioxide, water, and light yield glucose and oxygen. It creates an intuitive trap, suggesting plants strip carbon to build sugars and exhale the remaining oxygen as waste. Chemically, this is false. Reconciling the math requires a different perspective. We have to look at photosynthesis as a series of coupled reactions where the output oxygen must have a measurable, verifiable source. To deduce the actual molecular pathways of photosynthesis, we have to isolate the variables. We can do this by analyzing the foundational data from classical biological experiments. Understanding the true origin of atmospheric oxygen provides the necessary context for interpreting plant metabolism and planetary chemistry. Before we can track the exact origin of the oxygen, we must scientifically validate the core inputs of the photosynthetic system. We start with light. In the variegated leaf experiment, a portion of a living leaf is tightly covered with black paper to block all incoming photons, while the rest remains exposed to the sun. When treated with an iodine solution to test for starch, the results are absolute. The light-deprived sections show zero carbohydrate production, while the exposed cells are packed with synthesized starch granules. Next, we test the carbon input using Moll's half-leaf experiment. A living leaf is partially enclosed within a sealed glass tube. Inside the tube sits a small amount of potassium hydroxide, or KOH. KOH is highly reactive. It aggressively absorbs all ambient carbon dioxide from the air trapped inside the sealed tube. Even when the entire setup is exposed to full sunlight, the carbon-deprived half of the leaf fails the iodine test entirely. No starch is produced. Light energy and carbon dioxide are dual, non-negotiable prerequisites. Removing either variable completely halts the anabolic process. Knowing how plants synthesize solids does not explain what they release into the atmosphere. For that, we look at gas exchange. In 1770, Joseph Priestley constructed his famous bell jar experiment. He sealed a live mouse and a burning candle together under a glass dome. Within minutes, the candle extinguished and the mouth suffocated as the available air was depleted. Priestley then introduced a living mint plant into the sealed jar. The plant restored the air quality, allowing the candle to burn and the mouth to breathe. Biologist Jan Ingenhousz sought to refine Priestley's setup by testing the plant under strictly controlled environmental conditions. Ingenhousz submerged an aquatic plant and observed its behavior underwater during peak sunlight, and then again in total darkness. He discovered that the life-giving gas, oxygen, is only produced as physical bubbles when the plant is actively bombarded by solar radiation. In the dark, the outgassing stops completely. He noted a critical secondary detail. These oxygen bubbles only emerge from the green tissues of the plant. They never form on the roots or brown stems. Green plant tissue acts as a solar-powered air purifier, actively releasing oxygen as a direct physical response to light. Sunlight operates across a broad spectrum of wavelengths. How a plant discriminates between these wavelengths to maximize energy yield requires mapping the physics of light absorption. In the late 19th century, T.W. Engelmann utilized a glass prism to split a beam of white light into its distinct color spectrum. Engelmann projected this split spectrum directly across a microscopic filament of green algae known as cladophora. To measure exactly which colors drove the highest biological output, Engelmann flooded the slide with aerobic bacteria. These microorganisms naturally propel themselves toward high concentrations of oxygen. This graph maps the photosynthesis action spectrum. The bacteria distributed unevenly across the algae, clustering intensely at the blue and red light bands. When mapped against pigment data, these peaks align perfectly with the absorption spectra of chlorophyll A and B. The volume of oxygen released by a plant is directly dictated by the specific frequencies of light energy that its chlorophyll can physically absorb. We have established that specific wavelengths of light drive the system, and oxygen is the exhaust. But does that oxygen actually come from the carbon dioxide? In the 1930s, microbiologist Cornelius van Niel finally solved the paradox through comparative microbiology. Van Niel studied purple and green sulfur bacteria. These organisms undergo photosynthesis, but they do not produce oxygen. He observed that these bacteria do not use water in their chemical pathways. Instead, they utilize toxic hydrogen sulfide as their hydrogen donor. Because of this specific chemical substitution, these bacteria do not exhale oxygen, they excrete pure elemental sulfur. This is the redox equation for sulfur bacteria. Carbon dioxide plus hydrogen sulfide yields a carbohydrate, water, and sulfur. Now place the standard equation for green plants directly below it. The chemical logic is inescapable. If breaking hydrogen sulfide yields sulfur, then breaking water must yield oxygen. The oxygen we breathe is exclusively the byproduct of photolysis, the light-driven splitting of water. This entirely debunks the carbon dioxide misconception. This provides the fully balanced stoichiometric equation for photosynthesis. To synthesize a single molecule of glucose, a plant must physically oxidize 12 molecules of water, venting 6 molecules of oxygen gas in the process. The hydrogen stripped from that water is then coupled to reduce 6 molecules of carbon dioxide into the final carbohydrate structure. These two distinct chemical reactions map perfectly to the physical architecture of the chloroplast, a specialized cellular body first detailed by Julius von Sachs. The light reaction, where photons are captured to fracture water into oxygen gas, occurs exclusively within these membranous thylakoid stacks. The dark reaction, which utilizes that trapped energy to reduce carbon dioxide into carbohydrates, takes place in the surrounding fluid stroma. The process functions as a dual-phase thermodynamic engine, fracturing water to power the assembly of the biosphere's entire carbon foundation.